Dr. Kaeberlein, it's my privilege to introduce you to the group. Thank you so much for being here. Dr. Kaeberlein is a professor of laboratory medicine and pathology here at the University of Washington School of Medicine. His research interests focus on the biological mechanisms of aging in order to facilitate translational interventions that promote health span and improve quality of life. He has published more than 200 scientific papers and has been recognized by several prestigious awards, including Young Investigator Awards from the Ellison Medical Foundation and the Alzheimer's Association, the Vincent Cristolfo Rising Star and Aging Research Award, the Murdoch Trust Award, and the NIA Nathan W. Schock Award. Dr. Kaeberlein has been awarded fellow status with the American Association for the Advancement of Science, the American Aging Association, and the Gerontological Society of America, in addition to being the current CEO and chair of the American Aging Association, and has served on the board of directors for the Federation of American Societies for Experimental Biology and the American Aging Society. Currently, Dr. Kaeberlein is the founding director of the University of Washington Healthy Aging and Longevity Research Institute and the director of the NIH Nathan Schock Center of Excellence in the Basic Biology of Aging at UW. Dr. Kaeberlein, we're delighted to have you here with us digitally, sharing your knowledge and passion with us here at the Department of Medicine. I'll moderate your chat for you, and so folks in the audience can feel free to drop questions at any time, and we will go ahead and get started. Feel free to begin.
Great. Thank you very much for that kind introduction. And it's really a pleasure to be here. And what I hope to do today is kind of talk a bit about the biology of aging, why I think that it is an exciting and important area of biology and medicine to be thinking about. And for this audience, I really want to emphasize why it's important as physicians for you to be thinking about the implications of biological aging as you care for your patients. And the title of my talk is designed to be a little bit provocative, and that is targeting biological aging, a new paradigm for 21st century medicine. And I'll tell you what I mean by that as we go through. And then I was asked to complete these disclosures. So I do consult for a company called Trivium Vet that is developing rapamycin formulation for companion dogs. And you will see why that's relevant as we go through my talk. So I want to start kind of big picture. And just note that traditionally, biomedical research and clinical practice has been largely focused on individual diseases. And the set of diseases and disorders that are shown here are some of the major causes of death and disability in developed countries. And again, traditionally, the approach has really been to wait until people get sick with these diseases and then to either attempt to cure the disease or at least alleviate the symptoms. And I think one of the things that's important to appreciate about these diseases in particular is they all share at their root a single greatest risk factor. And it's not how much you drink or how much you eat or whether you exercise, it's how old you are. And so the basis of my research and most of the research in the field of geroscience is to really try to understand, what are the biological mechanisms of aging that cause aging to be the greatest risk factor for all of these diseases. And I think we can have a debate about whether aging causes these diseases. Personally, I'm convinced that the biology of aging does cause these diseases. But I think at a minimum, we all have to agree that aging creates a physiological state that is permissive for all of these diseases. And so what is it about that biology of aging that creates this physiological state that leads to the increased risk of developing all of these diseases at the same time? And if we can understand that at a molecular, biochemical, genetic level, we have the potential to actually target those mechanisms of aging to delay the onset and progression of all of these diseases at the same time. And so that's kind of the big picture of where I believe 21st century medicine is going. So I think of 20th century, 19th century medicine as trying to cure individual diseases. And 21st century medicine is really trying to approach kind of the root cause of why all of these diseases increase exponentially as we get older. And I think sometimes it's useful. I think probably everyone in this audience would agree that age is a big risk factor for these diseases, but sometimes it's useful to actually see the numbers. And so what's shown here are approximate relative risk factors that are often discussed and I think considered in the clinical community. I'm sure many of you, when you've had discussions with your patients, have recommended that they drink less or stop smoking or try to improve their diet. Those things are all real risk factors for disease. And these are three of the major killers in the United States. And you can see the relative risk. These are a little bit squishy numbers. It's surprisingly hard to figure out what the actual relative risk is for some of these factors, but these are within, twofold for sure. And you can see the relative risk for these major risk factors for disease, are somewhere between 1 and 2, sometimes as high as 3 or 4-fold increase in risk. Now, if we compare that to the increase in risk of biological or even chronological aging of about 30 years. And actually, these numbers are not squishy. So the CDC, believe it or not, is really good at keeping track of what people die from. And so you can come up with pretty good risk estimates for the effect of aging on these diseases. And so the same data is shown here, only with aging added. And again, this is approximately the effect of 30 years of aging. And the only point I want to make is that aging really is the predominant risk factor for these three diseases and many others. And it really dwarfs the common risk factors that most of us think about. And so, I might suggest that if you could have a discussion with your patients about what they really should be doing, maybe you shouldn't tell them to stop smoking or stop drinking. No, you should still tell them that. But maybe what you should really tell them to do is to stop aging. And I can't see most of your faces, so I can't tell if that got a few chuckles or not. But I'm going to guess it did. And I think probably the reason why that sounds funny is because we sort of all assume that aging is inevitable, right? That it's the accumulation of damage with time, and there's really nothing we can do about it. And I believe that's true for chronological aging. I'm not a physicist, but I'm pretty sure we can't manipulate time yet. But as a biologist who studies aging, what I want to tell you today is that we are really close, if not already there, in terms of being able to modify biological aging in people. And we do it all the time in laboratory animals. And that's kind of the take-home message of my talk today. And so we can start to ask, given that, what might that mean? And so this is a graphic that was published a few years ago in a review I wrote for Public Policy and Aging Report. This is really based on work that was done initially by a demographer, J.L. Shansky and his colleagues, where again, because the CDC is so good at keeping track of what people die from, it's pretty easy to ask the question, what would the impact be on population level survival and life expectancy from curing individual diseases? And so what's shown here is the impact of curing cancer or heart disease or both of those for life expectancy for a typical 50-year-old woman in the United States. And this sort of surprised me the first time I saw it, that it turns out that if you could cure all forms of cancer, a typical 50-year-old woman would only get 3 to 4 years of added life expectancy. And the same thing's true for heart disease. And even if you could cure both, you're still at less than a decade of increased life expectancy for a typical 50-year-old woman. Now, why is that? It's because all of those other diseases of aging are unaffected, right? So even if we fix one thing, all of these other diseases continue to increase exponentially. And if cancer doesn't get you, diabetes, Alzheimer's disease, kidney disease, something else is going to. Now, if you compare that to the impact of slowing aging, and here I'm sort of modeling this on what we routinely do in laboratory animals now, the added value there is much greater proportionally. In addition, those extra years are predicted to be spent in relatively good health, right? And that's what the green is supposed to illustrate here. Because you have delayed the onset and progression of all of these diseases of aging, or at least most of them simultaneously. Now, obviously, we don't know for sure that this can be done in people, and we don't know that the extrapolation from rodents to humans is going to be linear. So this is a projection of what might be possible given what is now routine in laboratory animals today. And again, I want to just re-emphasize this point that in the field, we talk a lot about lifespan, and we measure lifespan or life expectancy quite a bit. It's a nice quantitative thing that we can do statistics on. And it's useful to know whether or not an intervention that we think is slowing aging is extending lifespan. That's the prediction, right? If you slow aging, you should increase population lifespan. But really the goal is not so much to focus on lifespan, is to maximize the period of life that is spent in good health, free from chronic disease and disability. And so the concept here that we talk a lot about is health span. Again, that period of life where you're not suffering from one or more age-related diseases. And the goal is to really maximize that period of health span where individuals are living life and have a good quality of life, free from disease and disability. So there's just a couple of big picture take homes I want to present before I jump into the meat of my talk. So one is, I think, when people like Matt Kaeberlein get on Zoom or get up in front of you and start talking about slowing aging, I think, especially to a general audience, it kind of sounds like science fiction, right? I mean, this is a theme that we've seen in the science fiction literature sort of since the beginning, and I just want to make the point that this is not science fiction. This is science fact. This is happening right now, and in fact, aging really is just biology. And I think there are a few ways to illustrate that and appreciate that. But one really easy way is just to look across nature, right? If you look across the animal kingdom, you can see that evolution and natural selection have figured out how to modulate the rate of aging by many orders of magnitude. And so some of different species that age at very different rates are shown here, from laboratory model organisms like fruit flies that will age and die in a few months, to mice in a few years, to pet dogs, which I think about a lot. We're sort of all familiar with the idea that one human year is about 7 dog years, about. That's just the same as saying dogs age 7 times faster than people do, about. And then there are some really interesting animals that age much more slowly than people do, like whales that can live a couple hundred years, or clams that can live 500 years. There are also sort of what we think of as outlier animals, like this pink wrinkly guy here, the naked mole rat, which are naked mole rats, it turns out, are genetically pretty closely related to other rodents, but they age about 10 times more slowly. And so by studying them, we can try to understand, what is it about their biology that allows them or causes them to age much more slowly. And so I just want to leave you with the idea that really the only difference between these animals, it really can boil down to two major things, right? It's their genes. Clearly their DNA is different, and it's their environment. And that's just biology, right? So there's a real biology here that underlies aging, and we're starting to figure it out. In fact, we've made quite a bit of progress at figuring it out. So this is a graphic taken from a review article written a few years ago in Cell called The Hallmarks of Aging. And what's shown on the right-hand side is just a listing of the 9 hallmarks that these authors identified. And I'm not going to take you through them because that would take too long, and it's really not so important for my talk today. Many of these you've probably heard of. Telomere shortening has gotten a lot of attention, epigenetics, mitochondrial dysfunction. The point here is that the field has been able to identify these highly conserved mechanisms that seem to play a role in the biological aging process across every animal where aging has been studied. And so as we identify and understand what aging is at a cellular, molecular, biochemical, genetic level, that gives us opportunities then to actually try to intervene in a targeted way. And so in principle, any or all of these hallmarks of aging are targets where we could intervene either through lifestyle changes or through small molecules to have a positive impact on aging and age-related disease. And so I don't want to leave you with the impression that we figured it out. Aging is really complicated, and there's a ton that we still have to learn, but we've made a lot of progress, and we actually now have targets that we can modulate, and we can ask, what is the effect on aging in animal models? And so this is just a list of some of the approaches that have been taken that seem to work really well in the laboratory. So there are a variety of genetic models of delayed aging that in yeast and worms and fruit flies and mice seem to have the same effect of slowing the aging process, increasing lifespan, delaying functional declines that go along with aging. And then there are a variety of interventions that I sort of put under either lifestyle interventions or drugs, small molecules. Dietary restriction, probably most of you have heard of. That's sort of the classic intervention for modulating aging in laboratory animals. But now we have several small molecules that seem to be quite effective, actually, at also affecting the aging process. And I'm going to talk a bit more in the next 15 minutes or so about one small molecule, the drug rapamycin, and its biochemical target, which is mTOR, which stands for mechanistic target of rapamycin. So we were not very inventive when we named mTOR. And so what's been seen is that both genetically, knocking down mTOR signaling in each of these model organisms, or pharmacologically, treating them with rapamycin, increases lifespan and broadly delays the functional declines that go along with aging. And we know actually quite a bit about how that's working. So what this graphic illustrates is kind of from a big picture perspective, the way that mTOR is affecting aging. And so mTOR exists in two complexes, mTOR complex 1 and mTOR complex 2. mTORC1 represents mTOR complex 1. That's the target that rapamycin inhibits. And so one of the nice things about rapamycin as a drug is it's extremely specific. So it's an extremely clean drug. As far as we know, it really has one biochemical target. That's a protein called FKBP12. And when it binds that protein, it inhibits mTOR complex 1. So mTOR complex 1 is really interesting and important. So it is, in some ways, you can think of it as the sensor that detects the environment, in particular growth and nutrient cues, and then helps the cell and the organism to kind of make the decision. Is it the right time to grow and reproduce? Or is it better to kind of shut down cell division and become stress resistant? And you can imagine that nutrients are really important when a cell or an organism is trying to make that decision. So mTOR directly senses amino acids. It's also responsive to things like growth hormone and IGF-1, so growth promoting hormonal cues, and it also senses stress signals, and it really is kind of a central decision node in the network that helps the cell decide whether to grow and reproduce or not grow and reproduce. And what's been observed is that with aging, in many tissues, there is a continued activation of mTOR that then leads to many of these hallmarks of aging. And what rapamycin can do is it can down mTOR, delay these age-related changes that we think lead to many of the functional declines and diseases of aging. And to some extent, that means rapamycin is working as a mimetic of caloric restriction. It's kind of tricking mTOR into thinking that nutrient levels are low. And that has the effect of delaying aging and targeting the hallmarks of aging. And this is obviously extremely complicated, and I'm giving you sort of the 2-minute version, but a lot is really known about how these downstream targets of mTOR are impacting the aging process and how rapamycin seems to be delaying it. So I want to talk a bit about what we know in the literature about some of the effects of rapamycin. So rapamycin, as I mentioned, is a specific inhibitor of mTOR. It's the most effective longevity drug identified to date. The effects on lifespan vary by organism and by dose, as you might imagine, but it can increase lifespan up to about 30%, average lifespan, and up to about 30% in mice. One of the things that I think surprised a lot of people when this was first discovered, but now has sort of become commonplace, is that you can start treating mice with rapamycin in middle age and still get essentially the full benefits or close to the full benefits in terms of effects on lifespan and effects on age-related functional declines. And that's really important if you're thinking translationally, right? If we really ever want to get out of the laboratory and into the clinic, we probably don't want a drug that's going to impact aging that you have to start giving to teenagers, right? First of all, they're not going to take it. And secondly, the risk associated with taking any drug from the time you're, in your early 20s till the end of your life is much greater than maybe an intervention that you can start giving to people when they reach middle age, when they really start to show these functional declines that go along with aging. The other point I want to make about rapamycin is in the studies that have been done in mice, particular, where you can really look broadly across many organs and tissues, it seems to be the case that rapamycin is broadly improving health outcomes during aging across pretty much every tissue where this has been studied, including very potent effects in brain, heart, immune, muscle, oral cavity, kidney, reproductive tissues. So it really seems to be that it's affecting the aging process throughout the entire animal and not just affecting one specific age-related disease. And I'll show you a little bit of data for a couple of these coming up. So, you might ask yourself, given that, why aren't we all taking rapamycin? And I think the one obvious answer is we don't know for sure that it's going to have these effects in people yet. But I think also there are some real barriers when people like me start talking about translation of rapamycin for use in older individuals of normal health status to have effects on aging. One is that probably those of you who know anything about rapamycin, in your minds, it has a bad reputation, because I think probably most people listening to this talk come from the clinical perspective. And rapamycin has been used clinically. It's been FDA approved for a couple of decades. It's been used clinically primarily in organ transplant patients to prevent organ transplant rejection. And it's given to those patients at high doses in combination with immunosuppressants. And in that context, rapamycin does have a long list of side effects. Some of them are not particularly life-threatening, but they're not great. So many people get mouth sores, anemia is a common side effect, high triglycerides, something that looks kind of like diabetes, defects in wound healing. And of course, since it's used as an immunosuppressant, there's a big concern that giving it to people who are not don't have organ transplants, that it might increase their risk of infection. So it's got kind of a bad reputation clinically. I just want to make the point today that while those side effects are real, they are in sick people taking high doses of the drugs. And the data that we have, which is not huge, but it's growing, on relatively people of normal health status taking lower doses of the drugs, I think are pretty compelling, that the side effects pretty much go away, at least the ones that most people would be concerned about at lower doses. And this makes sense, right? Every drug has a dose response. If you take enough aspirin, it will kill you. But I don't think a lot of times this is appreciated when we talk about something like rapamycin. So I think there's reason to believe that this might be doable in relatively healthy older people. But having said that, this concern about side effects, I think, is a general concern when we start thinking about translation of interventions from preclinical biology of aging into the human population. There's a very low tolerance for risk when you're talking about treating older people who don't have overt disease. And I don't have time to get into this today. I think it's actually a really important and interesting discussion. Should we be willing to tolerate a higher level of risk if we might be able to give people 10, 15, 20 extra years of healthy life? I would say yes, we should, but there is a real barrier to getting people to consider an appropriate level of risk in older people who don't have overt disease. Okay, so I'm not going to talk about that anymore, but I think it is. It's a challenge certainly, and it's worth a discussion. So having said all that, one of the things that we sort of set out to ask was, might it be possible to get the same benefits, or at least some level of these benefits that have been seen from rapamycin in the context of aging, from short-term treatments, where you would expect the risk of side effects to be greatly reduced. And so I'm just going to show you a few cases where now this seems to be the case, where we can treat old mice with rapamycin for a short period of time and see, in many cases, quite dramatic improvements in function, which was a little bit of a surprise, I think. If you had asked people 10 years ago, if we target aging, how is that going to work? Most people would say, well, it's going to slow the damage, right? It's going to slow the decline. What's been seen with rapamycin is actually, in many cases, you can restore function that's already been lost, at least in mice. And so one example of this is work looking at cardiac function in aged mice. This is actually data from a paper that Peter Rabinovich's lab published. Peter is in the same department that I'm in here at UW, where they looked at the effects of 10 weeks of rapamycin treatment on age-related cardiac function. And so the way the data is presented here, there are young mice that got the control treatment, there are old mice that got the control treatment, and then there are old mice that got rapamycin. And the white bars are before the treatment period, so before the 10 weeks, and the black bars is that group after the 10 weeks. And so the couple of measures are shown here: left ventricular mass and E to A ratio, which is if you don't know what that is, it's sort of a measure of left ventricular function. And so what you can see, if you just compare the young and the old controls, is there is an increase in left ventricular mass with age, and there is a decrease in left ventricular function with age, okay? And that's kind of independent of this 10-week treatment period, because they're the control group. One thing I'll just mention is that at least for left ventricular function, just during that 10-week period in the old group, you can actually see a further decline in function. That kind of makes sense. This is the point in life where you're kind of going off the cliff. So then what happens in the mice that are going to get rapamycin? So before they see rapamycin, they look just like the old control group. That makes sense. They haven't seen the treatment yet. After 10 weeks of rapamycin treatment, their left ventricular mass is almost back down to where the young mice are, and their left ventricular function is back up to where the young mice are. So at least for these measures of cardiac aging, 10 weeks of treatment with rapamycin in 20-month-old mice is enough to take the old heart and make it look and function like a young heart, okay? And I'll just mention 20 months of age in mice is, I mean, it's hard to know exactly what that means in human terms, but it's probably around 60 to 65 biological years of age. So this is pretty compelling, right? That rapamycin 10 weeks can have can cause an improvement in age-related heart decline. Very similar sorts of effects have been seen for age-related declines in immune function in mice. So this is a study from Pan Zheng's lab at Michigan, where they were looking at influenza vaccine response. And the way this study was designed, similar in design to the one from the Rabinovich lab I just showed you, old mice, in this case 24 months, so even older, they get rapamycin for 6 weeks or control. Then there's a 2-week washout period, and then they get a vaccine to influenza, to H1N1 variant. And then they wait 2 weeks, and then they challenge them with influenza to ask, did that vaccination work, okay? And so this is the survival after that 2-week influenza challenge. And so I'll just walk you through this. So one thing to note is what happens if you're a young mouse, okay? If you're a young mouse and you don't get immunized, and then you're challenged with influenza, you'll die in about 8 weeks. All of the mice are dead, or sorry, 8 days. All of the mice are dead in about 8 days. So this is a lethal dose of influenza in a non-vaccinated background. If you're a young mouse and you're immunized, you're 100% protected, at least out to 2 weeks, right? The time course of this experiment. That makes sense, right? The young immune system is functioning the way it's supposed to. It responds to the vaccine. When I first saw this, this really struck me. If you're an old mouse and you get vaccinated, there's about a 70% chance that your immune system is not going to respond to that vaccination, and you will die when you're subjected to the influenza challenge. So this is, I think, a pretty clear indication that in mice, the response to influenza vaccine by the immune system declines quite dramatically. But if you're an old mouse that got 6 weeks of rapamycin, that's the dotted line up here, again, back to youthful levels, 100% protection from the vaccination against the subsequent influenza challenge. So at least for vaccine response, it seems to be the case that rapamycin can reverse the age-related declines in immune function. And what's pretty exciting is there have been 2 phase 2 trials now published showing similar effects from a rapamycin derivative in older people who do not have overt disease. So both improved flu vaccine response and apparent protection against all respiratory tract infections over the next season. So, it's still a little bit early, but I think now hundreds of people have been treated with low-dose mTOR inhibitor in these trials, minimal side effects, and evidence for restoration of immune function in healthy older people. And then this is work from my lab. This is work that John Ahn who was a former graduate student in the lab, did. John was a DDS PhD student in the lab. It's my understanding that John is now in consideration for a tenure track position in the School of Dentistry here at the University of Washington. If there's anybody from the School of Dentistry listening, you better hire this guy. He is a rock star, and you will be sorry if you don't get him on your faculty, because someone else will. So John came to me with the idea that we could take the geroscience perspective, which is understanding how the biology of aging affects age-related diseases, and apply it to the oral cavity. And so he developed a mouse model of periodontal disease, showed that indeed during normal aging, mice get the same clinically defining features of periodontal disease that people do. This is a very relevant disease in humans. About 2/3 of people over the age of 65 will get periodontal disease, and people who have periodontal disease are at higher risk of diabetes, dementia, and heart disease. So it seems to be linked to other clinically relevant diseases of aging. So John had the idea that not only could we potentially model periodontal disease in mice, normally aging mice, not a designed model where we take young mice and do something to them, but normally aging mice, we could ask the question, does something like rapamycin have an effect on periodontal disease? And I wouldn't be showing you this slide if it didn't. We saw very similar data to what I've already shown you for heart and immune function, where we could reverse all of the clinically defining features of periodontal disease from 8 weeks of treatment with rapamycin. That includes regrowing periodontal bone. So these are micro CTs that John took of the same animal before and after rapamycin treatment. And you can see that in this case, this space here where the bone has receded from the teeth is actually filled in with new bone. So we saw regrowth of periodontal bone. We saw massive suppression of inflammation in both the gums and the periodontal bone, and a reversion of the oral microbiome, which changes with aging in a way that people think is contributing to the disease, back to something that looks more like a youthful composition. And I'm not going to show you all that data. This was recently published in eLife if you're interested. But this is another case where short-term treatment with rapamycin seems to have pretty potent effects at not just slowing functional declines with aging, but actually reversing them in mice. And then what about lifespan? This is also work from my lab from Alessandra Bito, who's an acting instructor in lab medicine and pathology, where we ask the question, if we just look at lifespan, what is the effect of short-term treatment with rapamycin? And so in this case, the mice were 20 months of age at the start of the treatment. We gave them either a vehicle control or rapamycin for 3 months, and then we stopped the treatment, put the mice back on the shelf, and let them live out the rest of their natural lives. We had a couple of different doses and different groups broken out by sex, but I'm just going to show you one of them. This, in all honesty, is the one that gave the biggest effect, which is why I'm showing it to you. But if you're interested in digging into it, this is all published too, so it's in this eLife paper from a couple of years ago. What's interesting here is the mice that saw rapamycin had a 60% increase in life expectancy after the treatment. So this is the survival curves from the end of the treatment period, end of that 12-week period, to the rest of their lives. So the mice that saw rapamycin had a 60% increase in life expectancy. One of the things that's interesting about that to me is the absolute magnitude of the life extension is actually bigger than the amount of time that they saw the drug. Right? And so if we think about biological mechanisms where the effect on life expectancy is going to be longer than the treatment period, it's interesting to kind of think about what types of mechanisms might lead to that. And I'm going to leave you with that and let you speculate on that. We have some ideas, but I'll tell you, we don't completely know, but it's interesting. The other thing I'll mention is obviously we don't know that this would extrapolate to people, or if it did, that it would be linear. But if it did extrapolate linearly to people, that would be about 2 decades for that 50-year-old woman that I alluded to previously. So pretty substantial potential increases in life expectancy from transient treatments with rapamycin. Okay, and since we're all dealing with COVID-19, I've been in my office about 4 times in the last, I don't know, 8 months or something. I am in my office today, as you can see. We're all sort of in the middle of this global pandemic. I thought, especially given that I showed you the data on immune function in rapamycin in mice, that it's worth just briefly discussing potential impacts for infectious disease and particularly COVID-19. And I think it's worth appreciating that COVID-19 is a disease of aging, not so much your risk of getting infected, but once infected, whether you are going to have a bad outcome, which I would define as being hospitalized and or dying, that is age associated with essentially the same kinetics as Alzheimer's disease, all-cause cancer, heart disease. So there is a real aging phenomenon here. And I think people have speculated about why that is. The things that I've heard are the elderly are frail and less resilient. They have comorbidities, which we also know increase the risk of bad outcomes from COVID-19. And they have a weaker immune system, which is absolutely true. Just like in mice, our immune system stops functioning as well as we get older. The point I want to make is these are all driven by the biology of aging, right? That the reason why older people are frail and less resilient, have other age-related diseases, and have senescence of the immune system is because of the hallmarks of aging or the biological mechanisms of aging. And this is important in the context of a vaccine. I know we are all super excited that the vaccines are rolling out. This will be immensely important moving forward, but they're not going to fix all of our problems. We accept that sometimes 500,000 people worldwide will die every year from flu, even with vaccines. And I think there's a possibility we may end up accepting the same thing with COVID going forward. We don't know yet, but it's a possibility. So vaccines will help, but they're least effective in the people who are most vulnerable, the elderly, and they may not provide broad protection as if viral subtypes arise. And so I think we should be thinking about other strategies, and I would certainly advocate for a geroscience strategy to maybe help improve the ability of the elderly, if they get infected, to fight off those infections. And so some colleagues of mine, actually, this just came out a day or two ago in Lancet's new aging journal called The Lancet Healthy Longevity, kind of lay out what we think of as a roadmap for how you might test whether rapamycin or a rapamycin derivative could be effective at reducing severe outcomes from COVID-19. So if you're interested, certainly would encourage you to check that out. And I think it's worth, at least thinking about what that might mean. We might see something that looks like this go to something that looks like this. And so this is a very simplistic, just if we assume rapamycin boosts immune function by 20 biological years, that's how I did this, right? So certainly there's no real great data to say that's what would happen, but it might. And so I think it's worth thinking about, this kind of a strategy, and especially in the context of COVID, what it might mean. And I'll just mention, in a yet to be published third clinical trial, which actually didn't meet the FDA endpoint, unfortunately, what they did see was that the mTOR inhibitor was really, really effective against coronaviruses. So there is, I think, a pretty good reason for COVID in particular and influenza to think that this might be a viable strategy. Okay, so I'll just summarize what I've told you so far, which is that the biology of aging is being solved. We're making a lot of progress. That I think that this is a more effective approach than waiting until people are sick and trying to cure their diseases. I hope this becomes common practice in medicine in this century. We've got targets, molecular targets in the hallmarks of aging. We've got interventions. They're the first generation, better ones will come along, but I think there's reason to believe that the first generation interventions like rapamycin have the potential for clinical relevance. And that we should be thinking about this both in the context of health span broadly, but also specifically in the context of COVID and other infectious agents that lead to respiratory tract infections in the elderly. And so now I'll just spend the last 10 minutes or so talking about this bigger question in my mind, which is, how do we start to bring some of what we've learned about aging out of the laboratory and into the real world? And there are lots of strategies that that people are taking. I'm going to tell you about a strategy that a large group of us here at the University of Washington and around the United States are taking, which is to really try to explore whether companion animals and pet dogs in particular can be a bridge, right, from the laboratory to the real world. And I have, you'll notice this kind of double-headed arrow here, because I really want to make the point. This isn't only about what we can learn from companion animals and translate to human aging. I think that there is intrinsic value in being able to increase the healthy longevity of our pets, because so many people care so deeply about their pets. People love their dogs. I love my dogs. And I think if we're able to improve health and longevity in our pets, that has intrinsic value for quality of life for people as well. This is part of a large project called the Dog Aging Project. So there are really two goals to what we're trying to accomplish at the Dog Aging Project. The first is a longitudinal study of aging, and this is purely observational, right? So the idea here is to follow a large number of companion dogs, pet dogs, throughout a significant portion of their lives and identify the genetic and environmental factors that are most strongly correlated with health outcomes, with healthy longevity and with specific disease outcomes in pet dogs. And then there's the second part of the Dog Aging Project, which is, can we do something about aging in pet dogs? Can we identify interventions from the preclinical world that actually have an impact on slowing aging in pet dogs? So why pet dogs? There are lots of reasons. This is just a short list. Dogs age very much like we do, but they do so more rapidly. I already sort of alluded to dog years and human years. The implication of that is that we can actually study aging in a feasible time frame in pet dogs and get to strong correlations and eventually to hypothesis-driven mechanistic experiments. Dogs have this really cool genetic architecture where we've got all of these purebred dogs that are, you can kind of think of them almost as inbred mouse strains. But on top of that, you've got this large mixed breed population, right? Which is genetically heterogeneous and diverse. Dogs also have really broad phenotypic diversity. So for almost any phenotype you think about, dogs are going to be more diverse than people, right? Think about body size and the difference between a Chihuahua and a Great Dane. And it's that combination of genetic architecture and phenotypic diversity that is really powerful for mapping genetic variants that might be causal for phenotypes, in particular phenotypes of aging is what we're most interested in. The big one I would say is that dogs share our environment, right? So probably more than any other animal, maybe with the exception of cats. I don't want to upset any cat people who might be in the audience. Dogs live in the human environment, and that's really important, that environmental diversity. And what these graphics over here are meant to capture are, just like in people, there are rich dogs. So I don't know if you can see this on Zoom, but this is the doghouse. That's the dog. There are poor dogs. There are dogs that live healthy lifestyles. And then there are dogs that have, maybe questionable lifestyles. I don't know if you can see that. I did not pose this dog. This came off the internet. This dog has a cigarette in its mouth. Dogs don't smoke, obviously, but they do breathe our air, right? And so we will get information about air quality and water quality. That's another thing to consider. Most people don't give their dogs bottled water. So the dogs are drinking water out of the tap, and we'll get that information just from zip codes. So that environmental variability is really important, right? To understand, what's important for aging in the real world. And we just can't capture that in the laboratory. It's a socially relevant animal model that engages people in citizen science. So all of our participants, the owners, are citizen scientists who are helping us gather data for our study of aging in dogs. And as I've already alluded to, our hope is that we will be able to improve the quality of life for the pets and their owners. Okay, so this is the structure of the Dog Aging Project. The large longitudinal study, the biggest group is what we call the Dog Aging Project PAC. Our hope is we'll eventually get to 100,000 dogs in the PAC. We're at over 28,000 right now, so it's a good start. And to be in the PAC, all owners have to do is nominate their dog through the website and then complete a pretty extensive, so I'm not going to lie to you, it'll take some time, a pretty extensive what we call health and life experiences survey that really tries to capture in detail the environment that dog lives in and its health history. In addition, the owners are asked to upload electronic medical records so that we'll also have, veterinarian-based medical records for each of these dogs. That's all people have to do to be in the pack. And then from the pack, a subset of dogs will be selected for higher resolution study. So there's the foundation cohort, which currently we're funded to have 10,000 dogs in that group. Those dogs will get their genome sequenced. So we'll get 10,000 genomes as part of this project. And then within that, we have the precision group, which we think of as the omics group. So these are the dogs that we're going to get really high resolution information on every year. So every year, the owners will bring their dogs into their veterinarians. We will get veterinary data from the exam, serum metabolomics, blood epigenome, fecal microbiome annually, and follow those dogs as they go through the aging process. And this will really be a systems biology, right, question to try to disentangle what are the signatures that are most relevant for health and specific disease outcomes during aging. We also have a centenarian group, which really just means dogs that live a lot longer than they should, right, based on their size and breed. Can we identify factors associated with those dogs, either in their genomes or in their environment, that are associated with their exceptional longevity? And then the clinical trial, which is called TRIAD, Test of Rapamycin in Aging Dogs, which will be a study of 500 dogs to ask the question, does rapamycin slow aging in dogs? So this is the pack. As I said, there are more than 28,000 dogs in the pack right now. We had a big milestone a few weeks ago. This is Sage, the 25,000th member of the pack, a German Shepherd mix, and I'm partial to German Shepherds. Sage lives in New Hampshire, but you can see here's the geographic distribution of the dogs in the pack. And as I mentioned, we get extensive health histories on those dogs, and also environmental and geographic data on those dogs. So it's really early. So we're just now starting to get the survey data in. So this is very, very early data, but I want to give you sort of a tip of the iceberg view of what we're going to be able to do. So this just shows you disease diagnoses. This is owner reported. This doesn't come from the veterinary records yet. Owner reported disease diagnoses across all of the pack. So as you can see, we capture a large number of diseases, many of which are age related. And we can already see things that we expect, right? So owner reported walking speed declines with age. Just like in people, dogs get slower as they get older. It's nice to see that. It's not surprising, but it is reassuring that we can tease that out just from the survey data. And there's some interesting things already emerging from the disease incidence data. So first of all, this is kidney disease. As we would expect from the human data, if there's a logarithmic relationship between risk of disease and age, for small, medium, large, and giant dogs. In this particular case, there's an interesting breakdown by sex, where in each of the size categories, male dogs seem to be at lower risk than female dogs. There's probably some interesting biology there. Cancers, again, a logarithmic relationship between risk of cancer and age. And if we break it down by size, not surprisingly, big dogs at all ages are at higher risk for cancer than small dogs. That makes sense. That's probably related to IGF-1 signaling, but I'm sure there are also other genetic and environmental components that contribute to that. We're really interested in multimorbidity. So how often do diseases of aging go together? Again, as you would expect, and as we know from people, with age, there is an increase in comorbidities. But once we get a lot of data, we can start to ask interesting questions about which comorbidities go together and create a multimorbidity network. So this, again, is extremely preliminary. I don't want you to worry about what is clustering here, but more the idea that as this data set develops, we're going to learn, at least in dogs with age, which diseases of aging go together. Sometimes it'll make sense. I think sometimes we're going to see surprises that are going to be interesting. And so just with the last couple of minutes, I will tell you about TRIAD, which is the clinical trial of rapamycin in aging dogs. Again, the goal here is really to test, does rapamycin slow aging in pet dogs? We're doing that because, as I've already mentioned, there's a huge body of preclinical evidence that rapamycin slows, or even in some cases reverses, declines that go along with aging. And we've got some preliminary data in dogs from a 10-week trial that we've already completed. This is published in Geroscience, and this was done actually at the VCA Specialty Clinic in Linwood. So some of you may be familiar with that clinic, where This was a double-blind, placebo-controlled, randomized clinical trial. No evidence for side effects over the 10-week treatment period, and at least some initial indications that we might be seeing the same effects on cardiac function by echocardiography that were in that paper from the Rabinovich lab that I showed you. So interesting preliminary data suggestive that rapamycin may reverse age-related cardiac decline in dogs. So because this is a study of dogs that are not diseased, right? There are some real challenges, which I've alluded to around doing clinical trials in that patient population, and where does the risk-reward ratio lie? So obviously, these are people's pets. Safety has to be our #1 consideration. I mentioned we've already done the first study. We're working at doses that we're confident are unlikely to lead to significant side effects. We have an animal welfare board and a data safety monitoring board. This is a double-blind, randomized, placebo-controlled trial. It's open to dogs of all genetic backgrounds, but they have to be at least 7 years old and at least 40 pounds. And that size weight range is because big dogs age faster than small dogs. So in this window, we are statistically powered to see an increase in lifespan in the 3-year study period. So that's why we have this age weight range for entry into the study. And we have 7 partner veterinary teaching hospitals. So all of these dogs will be seen at our partner veterinary teaching hospitals, not at private clinics. So this is the design. I mentioned it's a randomized placebo-controlled trial. Half the dogs get the placebo, half the dogs will get rapamycin. Every 6 months, they'll come into the clinic. So we'll get a veterinary exam, we get echocardiograms, we'll get the fecal microbiome, metabolome, blood chemistry, cognitive assessments, activity monitors, and of course, we'll be looking at disease incidence. So our primary endpoint is lifespan, but we will have a whole bunch of secondary endpoints that we think will inform us about the overall health of the dogs as they're going through the clinical trial and at the end of the 3-year period. So this is my last slide, and this is just kind of where we hope to get in 2021. So we'll get the first true longitudinal data from the Health and Life Experiences Survey, as every year that will go back to owners. So all the owners who have already completed it will do their 1-year follow-up. We've rolled out cognitive assessments to everybody in the pack. We anticipate that we'll have half of the foundation enrolled, half of precision enrolled, and 100 dogs in TRIAD by the end of 2021. And I hope that we can get the pack up over 50,000 by the end of this year. And so with that, I will encourage you, if you haven't already and you have a dog, to visit the website, dogagingproject.org. Click on this button that says nominate your dog. You don't have to be in the clinical trial, right? You can be in the longitudinal study, and you can contribute to what I think is going to turn out to be really exciting, interesting, and important discovery in the biology of aging that will help improve the health and longevity of our pets. And we've got a fantastic team, too many people for me to recognize. I will recognize Daniel Promislow, who's the co-director of the Dog Aging Project, also in lab medicine and pathology here at UW, and Kate Creevy, who is our lead veterinarian, who's at Texas A&M. And it's really Katie, Daniel, and I that kind of guide this ship, but it's a huge team at many different places of really fantastic people who are really getting this up and off the ground. So with that, I'll stop, and I've talked for a while, but it looks like we still have a few minutes, and I'd be happy to try to address any questions or comments that people have.
Well, first and foremost, thank you so much. That was a really excellent talk. A couple questions have already come in the chat, and I think first was a question sort of whether you might briefly discuss the specific molecular and physiologic mechanisms underlying how mTOR affects biologic aging.
Yeah, I'll try to keep it brief because I mean, this really would take an hour to dig into, but I'll just go back to this graphic here. So I think the first thing to say is at one level, rapamycin in particular is very specific, right? As far as we know, everything that it does is through inhibition of mTOR. But then downstream of mTOR, there are all these processes that mTOR regulates that seem to be really intrinsically important for the biology of aging. If I had to pick 3 of them, and these are tied together too. I think a lot of people think about autophagy as being really important. So autophagy is this lysosomal degradation pathway where sort of damaged macromolecules can be broken down and recycled into useful macromolecules, right? So some people call it the recycling center of the cell. So we know that mTOR is an inhibitor of autophagy. So when you turn down mTOR, either through nutrient restriction or rapamycin, you boost autophagy, and that leads to sort of a breakdown of damage and creation of new macromolecules. So that's one important role that seems clear, that this is an effect of rapamycin and mTOR inhibition that's important for aging. The other thing that I think is really important through mechanisms that are still being worked out is this effect of mTOR inhibition on chronic inflammation. So chronic inflammation, to my mind, is perhaps the most important thing that goes wrong during aging, and it's sterile inflammation for the most part. And through mechanisms that are still being figured out, when you inhibit mTOR in an old organism, you really tamp down these chronic inflammatory signals. And I think that's, if I had to guess, I think that's partly why we see rejuvenation in many cases with rapamycin, because the chronic inflammatory environment causes cells and tissues to no longer be able to function the way that they're supposed to. It's not that intrinsically those cells are broken, it's that they just can't do their job because of the environment that they're in. And by inhibiting mTOR, you kind of tamp that down, and it allows the stem cells to function the way that they're supposed to. So from a functional perspective, what you see is rejuvenation of function. That's my guess.
Along those lines, I think folks are asking, just from the clinical perspective, what are the doses of rapamycin that we're talking about in human trials? And then are you seeing an effect on IL-2 signaling or other sort of inflammatory markers with those doses?
Yeah, so the two phase 2 clinical trials that I sorry, that I mentioned, both used everolimus, which is a derivative of rapamycin, so it's a rapalog, but the biochemical mechanism is essentially the same. So the doses that were used in those studies that gave the best effect, the dose that gave the best effect on flu vaccine, and I think also respiratory tract infection, was 5 mg once per week. They went up to 20 mg once per week. They also tested sort of the dose equivalent of 20 mgs, but given daily. One of the interesting things that came out of that was that the side effects were much lower at the once a week dosing versus the daily dosing. And there's some thought that the side effects from rapamycin may be largely driven by trough levels. So if you don't let the rapamycin bottom out and get restoration of mTOR signaling, that's contributing to a lot of the side effects. But by going to once a week, that actually allows the rapamycin to be cleared. So you get this sort of cycling of mTOR inhibition and then normal mTOR signaling, and that might be important. That's a little bit speculative still, but I think most people who are thinking about clinical application of rapamycin and rapalogs in older people of normal health status. I'm trying to stop saying healthy older people. Again, this is a little bit of a digression, but I think it is conceptually misleading to talk about older people being healthy. Sure, they're healthy in the sense that they don't have overt disease, but they're not as healthy as they were when they were 30, at least functionally. So I'm trying to, that's why I keep saying, I know it's a little bit awkward, but that's why I keep saying normal health status. Anyways, that people who are thinking about the use of rapalogs in that context are mostly, as far as I know, thinking about this once a week kind of dosing protocol in around the 5 mg once a week range. But it's really early days, not enough data to know with any certainty whether that's the right approach or that's the right dose.
Got it. Thank you. I think that another question that came up sort of in I feel like in popular discussions around this, a lot of folks talk about insulin, calorie restriction, like you're talking about, and also metformin has been brought up as a potential therapy for aging. Do you have any thoughts on that?
Yeah. So I mean, metformin absolutely is probably the other drug that people would point to in the aging field as sort of at the same level as rapamycin. Metformin and rapamycin in the literature is really interesting. So in mice, rapamycin, rock solid, always works, extends lifespan, delays aging. Metformin doesn't increase lifespan in mice, most of the time at least, and its effects broadly on aging are less clear. So from a preclinical perspective, metformin is nowhere near as good as rapamycin. But if you look at the clinical data, it's really quite compelling. So in diabetics who have been taking metformin, it's clear that they do better than diabetics who don't take metformin for life expectancy and a variety of disease outcomes. And there's at least a little bit of epidemiological evidence that diabetics taking metformin might live a little bit longer than non-diabetics not taking metformin, which is interesting. And also, you can find indications in the literature that diabetics taking metformin are protected against other age-related diseases. It's not perfect, and you can find counterexamples, but certainly there is evidence in the literature that metformin may be affecting other diseases of aging and not just diabetes. And the challenge with metformin, is that we don't really know biochemically exactly what it's doing. Most people will point to AMP kinase as the mechanism for how metformin is acting, that it activates AMP kinase. But unlike rapamycin, metformin is a really dirty drug, and it hits dozens of targets in the cell. It's a electron transport chain inhibitor, right? It hits protein kinase A. So we don't really know how it's working. And so it's a little bit harder to put it right in the network, the aging network. That's the way I think of it, for genes and proteins that we think we understand how they're affecting aging and know exactly where metformin fits. But from a clinical perspective, a lot of people like metformin to target healthy aging for safety, and primarily, right? Because we know, there's, again, as I mentioned, there's a lot of concerns about rapamycin and side effects. We know a lot of people who've been taking metformin for a long time, and there's less concern around side effects. And so I think that that's why you hear it talked about maybe even more than rapamycin as sort of the first drug that might be tested in this context.
Well, thank you again for such an excellent talk on such an interesting topic. I really enjoyed this a lot, and I think everyone has as well, from judging from the comments. So thank you so much. I think the sort of core concept of conceptualizing of aging, biologic aging as disease, independent of chronologic age, it's sort of a super compelling thought, and it begs a lot of questions, like you said, that would take their own hour to sort of muse over. So thanks so much, and thank you everyone for tuning in for Grand Rounds, and enjoy the rest of your Friday afternoon.
All right, thank you very much. It's been a pleasure.
Thanks so much.